I want to tell you about a very serious facial condition that I developed a number of years ago while I was here studying biomedical engineering. It's a condition that affects quite a lot of young people. It affects their facial appearance, and in my opinion, it's nothing to be embarrassed or ashamed of, though it is quite serious. But if I may, rather than talking about it, let me show you a photo of someone who has this very serious facial condition. So it's called facial confusion syndrome, and it's named after the emotion of confusion. And pretty much if you have it, you just look really confused at all times of the day. But all joking aside, what was the reason I was so confused back then? Well, the reason I was so confused was because I didn't know what I wanted to do after I graduated from biomedical engineering. Now, looking around the room here, I do see a few mild cases of facial confusion syndrome. So some of you may be going through what I was going through when I was in your position only a few years ago. So what we're here to talk to you guys today about is why you should consider doing a PhD in biomedical engineering. So my name's Ben Ferguson. I'm a biomedical engineering PhD candidate in the ARC Training Centre for Innovative Bioengineering and the School of Aerospace, Mechanical and Mechatronic Engineering. So biomedical PhDs, to be clear, this will be after you graduate from your current course. You'll decide to either go out into an industry-related job where you'll be working at a biomedical company, or you'll decide to undertake a higher degree by research for which you'll be involved in a 3-year independent research project. Yeah. So interestingly, the overwhelming majority of undergraduates, so this was 99% of my friends when I was graduating here a few years ago, the overwhelming majority decide not to do a PhD after they graduate, and they will go immediately into industry. So they'll go to Stryker, they'll go to Cochlear, ResMed, Johnson & Johnson, but very few consider PhDs. Now, why is this? Well, after speaking with many of my friends at the time, and just many people about this generally, I found that there are 3 common reasons or perceptions about PhDs that discourage students from wanting to do one. So it's commonly thought that a PhD is just esoteric theoretical research of little practical benefit to society. You're going to develop a deep but narrow skill set, and that's going to limit your career opportunities. And finally, insufficient funding. How am I going to fund my PhD? What I'm here to tell you guys today is that, boom, these are misconceptions. And these misconceptions are what discourage many undergraduates from realizing a valuable opportunity. So what we're going to do today, we're going to go through each of these point by point and dispel these misconceptions. And how we're going to do that is we're going to show you actual examples of biomedical engineering PhD research projects going on right now at Sydney Uni. So I'll be talking a little bit about my research. These talented PhD students are going to be telling you about their brilliant research. And hopefully from that, we're going to firstly dispel these misconceptions, but we're also going to equip you guys with more information as you go forth in making decisions about your future career steps. So, esoteric theoretical research of little practical benefit. Allow me to tell you a little bit about my own research. So, right now, we as a human species are in a 3D printing revolution, and we're in a biological revolution. The amalgamation of these two is what we call tissue engineering. Tissue engineering is where you're regenerating living human tissue in the body that has become damaged or diseased. One of the key ways that we can do this is by 3D printing special biomaterials that you implant into the body, and these special biomaterials are going to help regenerate new tissue. So I'm not saying substitute the tissue with some artificial prosthesis. I'm saying regrow the tissue back to its original healthy state. So let me show you an example of this. Here is a 3D model of a real human jawbone. Now, say if this patient experiences some form of mandibular cancer that's going to need to be surgically resected, you can see that by surgically resecting this cancerous tissue, the patient loses a large volume of their jawbone. And because this defect is so large, it's not going to be able to heal back without some form of external intervention. So, to reconstruct the jawbone, what clinicians have done in the past is they'll transplant living tissue from another part of the patient's body and they'll try and implant that into the defect. So usually they'll take a fibular graft from the patient's leg and they'll bring that up and they'll try and shape it to the volumetric dimensions of that defect. But you can see this remains an imperfect solution because you've got to do a secondary surgery to harvest the living tissue. And then it's also difficult to take the existing bone graft and fit that to the dimensions of the defect. So it's an imperfect solution. Here's what a tissue engineer would do. A tissue engineer will take a CT scan of the patient and then using 3D computer modeling will design and fabricate a bioceramic tissue scaffold. And they'll implant that into the defect. And they'll attach a titanium fixation plate to stabilize the whole construct. And the reason why we use a bioceramic scaffold is that your bone tissue is a form of ceramic itself. So the body's going to readily accept this biomaterial. And what you'll see over time is that the surrounding bone tissue is going to grow into that scaffold layer by layer. And concurrently, as this bone ingrowth is taking place, the scaffold will dissolve away into the bloodstream. And then you remove the titanium fixation plate because it's no longer required. And what you can see is that you've regenerated the original bone tissue, you've restored the shape and function of that jawbone. So that's tissue engineering, regenerating new tissue. But hold on, let's go back for a second. So what does my research actually involve? How are we going to actually do this? Well, what my research involves is designing and optimizing these medical devices so that when we do implant them into the patient, we can be confident that it's going to bring about the best possible healing outcome. So when we're thinking about the design of these devices, what would we need to be thinking about? We could think about the 3D microstructure of the tissue scaffold. Different microstructures are going to affect how bone grows into the scaffold, and it's also going to affect the mechanical performance of the reconstructed jawbone when the patient's biting down on it, when they're chewing food. Or we could think about the position at which the fixation plate is attached. And as my latest research paper has shown, this substantially affects the function of the reconstructed jawbone, and it's going to affect the healing outcome as well. I'll just also say that I've shown you a clinical scenario of mandibular reconstruction, so reconstructing a jawbone. But the principles and methods developed in this research project can be readily extended to also reconstructing other areas of the skeletal system too. So it's not just restricted to the jawbone. We could also apply these same methods to reconstructing the spine or the femur and so forth. But this has just been an introductory view to my research, which I totally hope you think is jawsome, by the way. But hopefully what this has shown is that a PhD is not just esoteric, theoretical research of little practical benefit. Biomedical PhDs are unique because they're very applied. An engineering PhD is not the same as a science PhD. An engineering PhD is more about coming up with a solution to a problem rather than a science PhD, which is more about discovery of some phenomena. So it's more of a practical emphasis. And what you'll find is that researchers in the biomedical school work very closely with industry and clinicians so that the research that we're solving is actually addressing the medical issues of today. So what you'll find is that it's much more of a practical emphasis, and we're really wanting to translate this research into tangible, practical, real-world solutions. Okay, so a deep but narrow skill set is going to limit your career opportunities. Well, generally there are 3 career options after you finish a PhD. You can decide to join industry, like my brother Eric did. Eric found that many of the skills required for machine learning in an industry setting, he'd already technically mastered during his PhD at the University of Sydney. And so what this meant is that when he graduated from his PhD and was hired in industry, he was immediately hired as the head machine learning engineer at the company. If you don't want to join industry, but you want to pioneer entirely new industry, you can do so in the form of a biomedical startup. So Dr. Ali Fathi, during his PhD at the University of Sydney, invented an elastic hydrogel that you can inject into the body, and it's used for tissue engineering purposes. He ended up patenting this technology, and then he launched his biomedical startup, Tetratherix, where he's now commercializing this invention. So that's another option, is launching a startup. And finally, if you do a PhD, after you finish, you can continue in academia like Dr. Xiao Li, who was actually my tutor for tissue engineering a few years ago. And Dr. Xiao Li, she ended up joining the Colling Institute and becoming a chief investigator in the ARC Training Center for Innovative Bioengineering, where she continues to work on tissue-engineered solutions to osteoarthritis. So what you can see is that in doing a PhD, you're not necessarily restricting your career options. What you can see from those 3 examples there is, if anything, you're enhancing your career options after you finish a PhD. Okay, insufficient funding. Well, there is a raft of funding available for a biomedical PhD. Generally, the most common way of funding it would be through the Australian Government Research Training Program Award, which, if you look up on the website, the annual rate is $35,629. You can also supplement this scholarship with top-up scholarships. For example, the ARC Training Center for Innovative Bioengineering offers a $10,000 top-up. There's other top-up scholarships available, and you can also choose to tutor. You pull these funding sources together, and that's how you can fund a PhD. And I'll just also mention that these figures are tax-free as well. Okay, so there isn't insufficient funding. I hope that I've been able to dispel these misconceptions. What we're going to do now is I'm going to pass on to these talented PhD students who are going to show you a collage of different biomedical research projects. We're going to expose you guys to as many biomedical research projects going on right now. And then after that, we're going to have a Q&A where we're going to be able to engage with you guys. You guys can ask us questions. And but before I finish up, I'll just say this. As first-year biomedical students, think about what field of biomedical engineering you like, and then tailor your electives and your honours thesis topic to this. You might find that your honours thesis ends up leading in to a PhD. Talk to your tutors and professors. They are a wealth of information. Ask them questions about their research. Ask them questions about their experiences. They've gone through what you'll likely be going through, and definitely engage with them. And finally, Google the ARC Training Center for Innovative Bioengineering. Check out some of the awesome research that's going on in that training center, and you'll learn a lot from that as well. So that's all from me. Thanks so much for listening.
So hey everybody, my name's Sam. I'm a PhD student in the Biomedical Engineering School as well. So my PhD is focused on the development of a novel EEG device to monitor cerebral blood flow. My supervisors are Andre Keim, who you guys know, Jeffrey Rogers and Philip Borton. So following a stroke, a patient will come into hospital to have a CT scan to diagnose a stroke. After that scan, they may be given treatment to restore blood flow. However, they won't have another scan for up to 24 hours. During this time period, there's currently no device for the continuous monitoring of cerebral blood flow and brain activity. We're trying to develop a device that can fill this gap, and we're proposing that this device can provide real-time monitoring of treatment, real-time monitoring of disease progression, and detection of new events, and also can this device also potentially reduce the number of CT scans this patient will require. So this is the problem my PhD is tackling. My PhD started off with a hypothesis. Can we record cardiac electrical signals, or the ECG, across scalp electrodes to detect changes in cerebral blood flow? So EEG is basically the placement of electrodes across the scalp. It's usually used to record brain activity. I started off with a commercial EEG device called the Emotive Insight, you can see on the screen here. It's a portable device, wireless device that you can wear. You can use it to record standard EEG measures, as seen on the next little box here. Those are brainwaves from 5 electrodes. I then grabbed a few ECG leads and did some rewiring of this device, and I found that I could actually record the ECG signal across the scalp electrodes. This answered my first part of my hypothesis. I then, on myself, doing some self-testing, I created an algorithm to process these signals, and then I did a number of different activities that alter my cerebral blood flow, such as holding my breath, hyperventilation, drinking coffee, and doing exercise. This led to the development of a new method to monitor cerebral blood flow changes. I actually then filed a provisional patent in 2019 to protect this method. Next, I created my own prototype device using open source equipment. So the OpenBCI is this open source platform that does electrical and biosensing devices. So I grabbed this device online, and I got a headband from Priceline, girl's headband, and I fitted into a bunch of electrodes into this device, added a few ECG leads, and made my prototype, as you can see on the screen. I then started conducting a number of validation studies to further validate this method. I've just completed my first study last week. We recruited healthy volunteers to do a number of activities that alter cerebral blood flow, including hyperventilation, blood flow, breath holding, exercise, and also a cognitive task. Participants completed these tasks while wearing my headset. They also had their blood flow monitored by transcranial Doppler ultrasound, as you can see on this picture up on the screen now. Here's the participant wearing my headset device where I was cycling, and you can see on the screen behind him, his blood flow has also been monitored with ultrasound. So this study formed my first publication for my PhD and forms more evidence that shows that this method may be able to monitor blood flow changes. I also have another study in process as well. And I've actually created a startup company called Neuroflux that now owns this patent that I've created. Recently, a few doctors at Westmead Hospital have agreed to help me do a clinical trial to further test the safety and efficacy of my device. So that's really exciting moving forward. And that clinical trial will form my third publication of my PhD. So this is my PhD journey. You can see it's got quite a commercial component to it. And if all my results go well, I'd like to continue my startup company in the future. Thanks.
My name's Cynthia PhD. I just started my PhD 2 months ago with Greg Swanning in the Sydney Bionics Lab, School of Biomedical Engineering. The topic of my thesis is the restoration of natural blink after facial paralysis. So basically, I'm trying to develop an implant that will be able to restore the function of blink in people who have lost that function from some pathology. So some background on how we blink. It basically comes in 2 stages, which are innervated by completely different nerves, completely different muscles. You first have the LPS muscle, which is innervated by cranial nerve 3. That one is tonically active, so it stays active the whole time to keep the tension in the muscle, to keep your eye open. When you blink, that nerve signals to deactivate the muscle, and this other muscle, orbicularis oculi, which is innervated by cranial nerve 7, gets activated. And that one is a sphincter muscle. It circles the eye, and it contracts and causes a blink. So while there's a bunch of causes of facial paralysis, they all generally affect cranial nerve 7, which means that your eye stays open, but you're unable to blink. So generally, people's vision isn't affected, so they can still see through their open eye. But if you've ever tried to have a staring contest, you know how quickly your eye can dry out and how painful that becomes very quickly. So because blinking has such a strong impact on the lubrication of your eye and to keep everything moist. So without blinking, someone can quickly get corneal irritation from the dryness, and it can eventually lead to blindness if you don't manage it properly. Other than that, you also lose a protective reflex from blinking. So if you have a foreign object going towards your eye, there's no way to protect your eye. And you also have an asymmetrical face. And while that doesn't seem as important as the other issues, people have a hard time integrating in society when they can't blink, because your eyes are the window to your soul, and people don't really trust you as much when you have an asymmetrical face, and so people kind of withdraw from society when they have this sort of problem. So there's a bunch of current interventions that people are trying. The current gold standard is to just put a weight into the eyelid. So basically, a gold or platinum weight is implanted into the eyelid, and when that LPS muscle deactivates, the weight just slowly kind of helps the lid to close from gravity. And so while that works pretty well in some people, because the blink, it's quite slow, a lot of the time you don't achieve full eye closure. And so then you don't get the proper lubrication of the eye, and so a lot of the time, people still experience the corneal dryness and still have a lot of other complications related to their facial paralysis. So you have an incomplete blink. Another option is electrical stimulation, but that requires you to have all of the anatomy still intact. With the rise of skin cancers in our country especially, a lot of people are getting a bunch of tissue taken out from the area around their eye, and a lot of the time the facial nerve needs to get sacrificed, and that causes facial paralysis. So then there's nothing there that you can actually stimulate properly. Same for if people are paralyzed for a long period of time, there's nothing left that electrical stimulation can help. So it relies on intact musculature. The other option is to transfer something like muscle from that's already used in your jawbone or your temporalis muscle, and kind of use that to recreate your blinking muscles. But a lot of the time that results in quite an unnatural blink, and so it doesn't really solve those cosmetic issues, as well as not entirely recreating the complete closure. So our solution to this is the bionic lid implant for natural closure, or BLINK. So this is an electromechanical implant. It's active, so we can kind of tweak it to make sure that you get full eye closure. It's a completely new system, introduced into the face, so it doesn't rely on any of the musculature being intact. And we are putting a sensor so that it can detect when your other eye blinks, and so it can trigger you to blink at the same time, which results in a more natural sort of appearance. So how does it work? So basically, we harvest some tissue from the person, and a long sort of section, and we implant that into the eyelid. One side gets attached to where your nose bridge is, and the other side gets attached to a magnet inside the device. So when the device gets turned on, a large capacitor, like a battery, inside the device gets charged up. And then when we get the trigger to blink, the capacitor quickly discharges through a coil, which becomes an electromagnet, pulls back on that magnet, and then tightens the sling. So you can see from that picture, the sling, when it's in the inactive state, is loose in the eyelid, and then when it gets activated, it pulls the eye closed by going straight. At the moment, what I'm focusing on is changing a bench-tested device, which we've also done some cadaver work with, and transferring that from a resin print into something that can be used in an actual live animal study. So what Ben was saying before about how we're in the revolution of 3D printing, we're using ceramic prints, which are able to be implanted as the basis of the design. And with some 3D modeling and some using CT scans and things like that, we're able to make a device which fits the anatomy, and hopefully we'll be able to produce the full and complete and natural blink that we're working on. And that's my group that I work with. Thank you.
So I'm Jed Vanderwallen. I'm in my second year of my PhD, and I'm looking at personalized myoelectric sensors. So unlike the others, I'm actually in the sort of electronic side of biomedical engineering. But I also implement a bit of 3D printing, which makes it a little bit innovative, but also cool. So I'll just do a bit of project background. I've thrown a few words out. Myoelectric is one, and sensor. So let's start off by looking at what is myoelectric. So it comes down to an electrical signal which is generated by muscle in the arm. It's always going on. When you contract your muscle, it'll get bigger. And when you're doing nothing, it will just sort of stay around and have a small signal. So when you think of contracting a muscle, for example, in your arm, it's actually originating from your cerebral cortex in your brain. It travels down your spinal cord, out through a peripheral nerve, such as your brachial plexus, and ends up at several muscle fibers. Muscle fibers are cells, and many cells are stimulated at the same time. What we're actually looking at is the sarcolemma. This is the sort of muscle lining, the sort of fat around a muscle cell. And this signal, the electrical conductivity across the cell, which is influx and efflux of sodium and calcium ions, is what we're interested in. This is a quick setup I made previously, and I'm using large hydrogel electrodes, which are normally used. They're known as the gold standard. And when I flex my arm, you can see that I get a red signal, which is the raw signal. It kind of looks like a cardiac signal. It goes up and down. And then I have underneath a rectified signal, which a bit of signal processing makes it seem like it's kind of related to the force of the muscle. This is all very well. I've popped 3 electrodes on my arm, but I'm sort of measuring all the muscle activity. I don't know what muscle I'm recording. So if I put it on my brachial arm, I may not just be recording the bicep, but maybe the tricep and the side muscles as well. So my question is, what electrodes do I need to use? I can use surface electrodes. They don't need to be implanted. And I can also use implantable electrodes. So I'm looking at both. Why not? Make it more complex. Why is this important? As many of you might have realized by now, one of the big applications is prosthetics. Okay, so having an arm which is not your own and trying to control that through muscle sensors. Another one is also functional electrical stimulation, and this is very useful for spinal cord injury, where they don't have any ability to contract their muscle anymore. If we could bypass the spinal column, contract the muscle, but also record what's going on so we can optimize it and make it more physiological. Other ones are robotic surgery, and a big one is actually control of personal devices. Your phone, laptop, TV, maybe even a car. This is actually probably the largest market for myoelectric signals, and it's a huge amount of people who do have difficulties with muscle control. Of course, there's diagnostics and research as well. We can use further clinical studies, such as gait or biomechanics, and also testing the health of muscles from its electrical signals.
There are 2 wireless myobands that John wears very comfortably.
This is known as an osseo-integrated implant. He has 2 muscle electrodes around his arm, and he's trying to pick up an object. This is state of the art. It's pretty cool, and he can actually pick up and contract a specific object.
To get the best solution. And it's not the best solution just for him. I mean, he really feels strongly about being able to advance.
So the only problem with this is he's only doing one motion at the same time. He's not extending his arm and then rotating his wrist and then grasping. It's sort of this. And he's using 2 whole bands, which comes to 32 electrodes to do one task. It's great, but we believe we can do better. So we want to make the sensor more comfortable, not so bulky. We want to have better muscle specificity so we can know what muscle we want to be stimulating, and have a high signal-to-noise ratio, which means we want lots of muscle signal and not much noise, so moving or maybe cardiac signals or any other signals which can cause interference. The big picture is looking at both surface and implantable sensors, having connectors, having chips which do analysis to determine, for example, through machine analysis, what muscle is stimulating, so we can use it for bioelectrical applications. And I'll just give you a glimpse of what my project is looking at and some results I've achieved with 3D printing. On the left, you see a tattoo sensor where I'm printing conductive ink. In this case, what I was printing was a conductive polymer, a plastic. And on the right-hand side was printing conductive silicon, yes, conductive silicon, on a glass pipette to see if we can have different examples of both surface and implantable. So that's my project. I'm in my second year, so I have a lot of work to do. But the next step is getting things to work, getting past all these troubleshooting, and then getting some results. And that's me.
So hello everyone, my name is Dylan, and like yourself, I graduated from biomedical engineering maybe 3 years ago, and now I'm doing a PhD with Professor Chin Lee, and my thesis topic is computational analysis and optimization of coronary stents. So I had a look on CASP, and I'm aware that the course structure has changed since the time I was doing it, so now time you get to choose your specialization. So I guess the stuff I'm doing will fall into specialization in biomedical modeling and design. So they'll be using computational models to simulate complicated and possibly also expensive experiments in biomedical engineering for medical device design. So for example, the design of coronary stents. So what are coronary stents? So basically, they are these tiny metal meshes that you put in your arteries when they get a little bit occluded due to coronary heart disease. And coronary heart disease is caused by the buildup of plaques, as you can see on picture on the right here. So when the plaque build up, your narrow kind of get narrowed. And ultimately, this will lead to the complete blockage of your vessel, and this will cause heart attack. And in Australia, this actually accounts for 3 in 10 deaths, so making it quite a serious problem. So the treatment can be done by surgery, and we call this the percutaneous coronary intervention, or PCI. So this is where you insert a stent inside your occluded vessel, and then you use a balloon to expand it. The stent will remain in the vessel after the surgery to provide further mechanical support. So there are a few factors that will influence how the stent perform, or we call it the stent biomechanics. So of course, the stent design itself is one factor. So which stent you're using, what sort of geometry that you decide to use for your stent. Also, there are the biological uncertainty. For example, the geometrical profile may differ from patient to patient, and this will affect how the stent performs. So it's not one fits all kind of stent. The performance will differ from one patient to the other. Also, the surgical uncertainty may be on the placement of the balloon, whether if it's not in the center of the stent, then it will affect how it will cause the stent to undergo uneven expansion. So my topic is on robust optimization of stent. So that means to optimize the geometry of the stent in order to improve its performance such that those random uncertainties I just mentioned will have minimal effect. So we do this basically by simulating the stent expansion process, and then we build some mathematical models to predict how the stent will perform. And then we apply some optimization algorithm to improve the stent design. And computational models, so this is what I do. I build up a few, like the vessel model, so you can see 3 layers of artery, and then 1 layer of the plaque, and then I also drew a balloon in the middle and the stent itself. So on the side view, you can see the flow path actually gets quite narrowed. Cool. And then I'll just show you the simulation itself. So you put the balloon in the middle, and then you kind of expand it. Yeah. So that's my research in a nutshell. That's all from me. Hopefully it makes sense to you guys. Thank you.
Okay, hello everybody. My name is Matilda Longfield. I am a graduate of the Biomedical Engineering undergrad degree, but I actually now do my PhD through the Faculty of Medicine in the Central Clinical School and the Greg Brown Diabetes and Endocrine Lab. So my PhD was actually born out of my honours project, and essentially the question that I'm trying to answer is it possible to develop a point of care bedside test for predicting the healing outcomes of chronic wounds, with a focus on patients with diabetes mellitus who have these chronic wounds? So just to give you a little bit of background before I jump into what I'm up to now, the reason that we care about this project is essentially people with diabetes-related foot ulcers. The regular healing process has been interrupted. So regular healing is sort of a multi-phase process, and a lot of these phases overlap. Now, delayed healing occurs when one of these phases becomes interrupted. And the phase that we focus on being interrupted in diabetes-related foot ulcers is this inflammatory stage. And essentially what's happening is we're seeing this becoming prolonged, and as a result, the wounds do not progress through the normal healing trajectory, and they get stuck at this earlier inflammation stage. So patients with diabetes have approximately a 25% risk of developing a DFU, and this is for a number of reasons, but it tends to be related to the comorbidities that are linked to having diabetes. So things like mobility problems, vascular problems, something called peripheral neuropathy, which essentially means that they have nerve damage in their lower extremities. And so all of these things combine together to mean that people that suffer from diabetes are more likely to get these hard to heal wounds. Now, we have a few ways that we try and deal with these at the moment. My supervisor is actually involved in the development of the high-risk foot service, and they run a lot of that out of the RPA hospital. But essentially the aim there is they've got doctors, nurses, and podiatrists all trying to work together as part of a larger team in order to give a holistic care to patients with these kinds of hard-to-heal wounds. And essentially what that means is that they're not healing within a 3-month period. However, even with this interdisciplinary high-risk foot service care, we're only healing in about 80% of ulcers. Now that seems great, right? 80% of ulcers are healing, fab. But why aren't this further 20% healing? And that's where my PhD comes in, trying to figure out how we can identify this higher risk group of these ulcers that aren't healing. So I started focusing on this idea or this project during my honors. And my honors project, it was a bit more zoomed out. It looked at delayed wound healing or chronic wound healing just in general, so it didn't have this diabetes focus. But essentially what I was doing as part of my honors was trying to develop a microfluidic paper-based sensor that could work as a diagnostic for wound healing. So what did we come up with? We came up with the following in my year-long honors project, and essentially the way that this worked was we focused on a few key characteristics of wound fluid, and we tried to use those to identify whether a wound was going to have a higher or a lower likelihood of healing. So the ones that we focused on was where we focused on the pH of the wound fluid, we focused on the viscosity of the wound fluid, and through the literature review, it suggested that the bacterial content of the wound fluid would be of use. So there was a stream that looked at anaerobic bacteria and a stream that looked at aerobic. And the idea was that this paper sensor would hopefully give us an idea, using these markers, of what was going on within that wound bed that might mean that it wasn't healing. And it worked in the following way. You would drip wound fluid into the window at one end, it would wick across the device, and as such, color changes would happen as a result of pH changes. Now, this device was made of paper towel that we bought from Kohl's. It was made of laminate, and it was made of some very smelly red cabbage indicator that I made in my kitchen to the dismay of my housemates. So it was pretty basic, but the main focus of this was that it was affordable, it was accessible, and it was reliable. But the problem was, at the end of my honors, we weren't sure we'd actually hit that last mark, the reliable one. And that's where the Faculty of Medicine got interested. We all thought we can do better. So the following question got developed. What are the primary biomarker candidates that are able to predict delayed and non-healing outcomes in diabetic wounds? And essentially, the Greg Brown Diabetes Lab, they'd done some research into this idea before and produced these following papers. And essentially, they'd found a couple of markers that they thought could be of interest and potentially a little bit more robust than my paper and red cabbage indicator that I'd come up with before. So they onboarded me. They said, come, join us. Let's do some research together. And I swapped over into the Faculty of Medicine, and I joined the lab that's doing this excellent research in order to try and figure out whether we had some more robust markers that we could identify in order to do this predictive healing of these wounds. So how am I approaching this question now? I'm approaching it in the three following ways. Firstly, through systematic reviews of the literature. Now, these are quite similar to the reviews we've been talking about with you guys throughout this course, but essentially it's a strict methodology where we search out in all the literature and I try and find out what everybody else that is involved in this space is coming up with and it is finding. The second way that we're approaching this question is through the use of animal models of wound healing. For some people, this might be a little bit a bit more controversial, but essentially in medicine, animal models are quite a normal way to have a controlled method. So I'm gaining loads of really great experience in terms of diversifying my skills and gaining new skills in terms of running these animal models and project managing them, but then also the lab components that come afterward in order to analyze the results and what we've come up with. And then the final way that I'm approaching this problem is through human wound fluid investigations. And this is pretty cool. We're using actual clinical samples. Luckily, I don't have to collect them myself. A lovely nurse over at the RPA hospital does that for me. But essentially, we're being able to look at the actual wound fluid coming from these patients that suffer with these diabetic foot ulcers, and we're being able to see the difference between the ones that we follow to healing and the ones that don't reach that healing endpoint. And so again, I'm gaining great lab skills and optimization skills through analyzing these tissues and doing these investigations. So what are we aiming for? Essentially, we're aiming for a point-of-care bedside test that can rapidly diagnose diabetes-related foot ulcers. Why? Because the benefits would be that we could assist diagnosis of these wounds at an earlier stage. We could potentially predict the healing outcome, which would help inform our clinical decisions and inform us whether we should, focus on this wound because it won't heal with the state-of-the-art technology in order to avoid amputation, or whether with conservative management and standard dressings and debridement, that wound actually probably will heal and actually can be sort of left to do its thing. And additionally, if we could create a point-of-care test, the idea would be that we improve the accessibility of care by creating something that can help patients even if the doctor's not in the room, because it can give them an indication of how their wound is doing. And what could be the future? We could integrate this with iPhone technology in order to create cloud sharing, and that could mean that clinicians across Australia and maybe in the developing world as well could share the results of these tests and then arguably back-validate them so that we can know that we are effectively predicting based on the biomarkers we've chosen. So I can't finish without acknowledging all the wonderful people that helped make my PhD possible. This is my lovely lab, and thanks to my supervisors, Stephen Twigg and Danching Min, and my various collaborators, because PhDs aren't done solely on your own. Lots of teamwork involved and great collaborations too. So yeah, thank you. That's all from me.
All right. Hi, everyone. My name's Claudia. I'm a third-year PhD student, and it's probably going to show in my project, but I feel like a bit of an imposter. I'm not actually a biomedical engineer. I'm a chemical engineer by training, but I found my way into what I think is a really cool field that kind of sits at the intersection between biomedical engineering and a lot of other disciplines. So I hope that I can maybe show you a different side of what biomedical engineering could be. With my project. So I am a genetic engineer now, but more specifically, a bacterial genetic engineer. So what's really cool about bacteria is that, while I'm sure you know that in humans, our DNA is packaged up into chromosomes, bacteria actually have two different ways of storing their DNA. So most of it is found in a chromosome, like in humans, but they also have this really nifty way of storing little pieces of DNA in little circular forms called plasmids. So a plasmid is just a free-floating circular piece of DNA. It might encode for just one gene. It might encode for several genes. And why we care about plasmids is that bacteria can really easily transfer plasmids between different cells, like they can give them to other bacteria which means that we can see this like really rapid transfer of different features, different functions, different genes between bacterial species really rapidly. So for example, if you had a purple bacteria and the gene that turned this bacteria purple was encoded on a plasmid, if that bacteria comes into contact with another bacteria, it can transfer that piece of DNA to this other, this other species. And once that species has taken up a plasmid as well, it can then also start making this purple producing gene. So the existence of plasmid DNA is actually one of the reasons why we think antibiotic resistance can move so rapidly between bacterial colonies. Antibiotic resistance genes are usually plasmid encoded, and so are genes that might protect bacteria from like heavy metal toxicity, for example. So they're usually really useful genes that you'd find on these little pieces of DNA. Now, as a genetic engineer, the reason that I care about this so much is that we can hijack this system. So I spend my days designing, engineering, optimizing plasmid DNA that I can kind of encode specific functions onto and then transform into my bacterial species, and I can create bacteria that have different functions that they may not have in what you would find in the natural world. So my project specifically isn't actually a biomedical engineering project, so I'm not going to talk about it too much. But I guess I kind of wanted to show you a plasmid, my example of a plasmid on the screen, to kind of help you understand what a plasmid could look like or how plasmid engineering works. So I like to think of it as playing Lego with DNA, right? So you have all of these functional components, like your genes. You have switches like you would in an electrical circuit. So if you were thinking about building an electrical circuit, you might have an on switch, you might have a dimmer, you might have a functional unit like a light bulb, and when you assemble them together, you can control what that circuit does. And the same is true for genetic circuits or plasmids. We can have switches that can respond to environmental cues that can then cause genes to be turned on or off or have their expression changed accordingly. So my project, I'm building a plasmid that when I give it to E. coli, the E. coli will be able to firstly detect hydrocarbon gases in the environment and then turn blue so that we can use these E. coli as like a little gas biosensor. But what I wanted to do today is kind of keep it relevant to the biomedical sphere and maybe give you a few examples of how engineering of things like bacteria or viruses or plants could also be biomedical adjacent. So one of the really cool projects in my lab was this open source insulin project. So a team of undergrad students actually designed this plasmid that contained the gene encoding for a form of insulin. And they gave it to Bacillus subtilis, which is generally recognized as safe. And when this bacillus started reading this plasmid, it started producing insulin. What was really cool about this system is that the insulin wouldn't just stay in the cell, it then got excreted into the media that the bacillus was growing in. So we could engineer bacillus to produce insulin, scale up that process, and be able to make huge amounts of insulin really, really rapidly, really quickly, and at a really low cost. So you can produce a drug that's really expensive, generally speaking, and inaccessible to a lot of people who have diabetes, who might be financially challenged. So there's a really kind of cool way that you can take something like bacterial genetics and use it to help treat medical conditions. Some of the other kind of examples of how bacterial genetics could be used for biomedical engineering technologies. So there's some evidence that you could engineer bacteria to detect, target, potentially destroy neoplastic cancer cells in the human body. You can help control disease propagation through a community by engineering insects so that they are unable to pass on the disease-causing toxin, I guess. And another really kind of cool example that I think is quite relevant to the time we're living in now is you could potentially genetically engineer viruses to be not so virulent, which could allow them to be used to develop live vaccine production, but in a really safe way. And I guess going back to more maybe traditional genetic engineering ideas, in plant genetics, you could engineer plants to change their nutritional profile or change how their nutrients are absorbed in the human body. So you can kind of work at this interface between tweaking things in the environment to have better health outcomes for humans. And that is all from me. So thank you.
So this part of the session is where we want to engage with you guys, and feel free to ask us as many questions as what you want. And we're also privileged to have the head of school, Professor Greg Swanning, who's going to be moderating the Q&A session as well.
Thanks, Ben. Are there any questions from the audience to begin with? If not, I'll start off by asking the panel, what drove them to the decision to go into, to get a PhD instead of going into industry, which is possibly the more traditional way of after you graduate? Ben, how about you?
I'd had quite a positive experience doing my undergraduate honours thesis. So the supervisor who I'm doing a PhD with now, Professor Ching Lee, I had originally done my undergraduate honours thesis with him. And during that time, we had sort of developed a good academic relationship. And it meant that when it came time to actually, finishing the honours thesis, considering doing a PhD, it was definitely quite a smooth transition because I'd already developed a good relationship with my supervisor. And so it was quite a smooth transition between doing the honours thesis to then doing a PhD. So I definitely recommend when you're doing your honours thesis, think about, firstly, whether you like the content. I personally just really enjoyed the content of the subject matter. And then what it meant is it was sort of a great introduction into that field. And then when I wanted to take it another step further in doing a 3-year PhD, it was quite an easy choice to make.
Jacinta?
So I started working as a research assistant in Greg's lab in my final semester of uni. And I'd already done an internship the summer before at Claudia. And I really liked both sort of working environments. But I found that by working in the lab in research, I got to do some really hands-on sort of stuff. So and especially so I worked as a RA for the last 2 or 3 years. And I got to kind of expand the amount of knowledge I had in engineering, do a bunch of different things, see different companies, like so many things that I may not have been able to do in industry. And as a lot of my friends went into industry, a lot of them had a lot less opportunity to kind of run their own project and do the hands-on engineering that I really loved. So I found that the research suited me and what I wanted to do more than industry.
Cool. So for my case, when I was doing my internship during undergraduate study, I researched in 2 different research institutes, and I really enjoyed it. And I realized maybe going into industry, I wouldn't be able to have the chance to run project by myself. And also, I'm doing like a biomedical modeling now, which I think is quite trendy in biomedical engineering, because we need sort of simulation to simulate more complicated human body system. So that's why I decided to pursue, study more into modeling the biomedical device and stuff.
There were quite a few reasons why I chose to do a PhD when I did. I think I always wanted to do a PhD at some point, but I wasn't sure if it was what I wanted to go straight into after undergrad. And when it came time to start looking for jobs, I realized that a lot of the genetic engineering jobs that I wanted really required you to have a PhD. They required that extra level of training. And so I kind of saw it as a necessary step to progress in the kind of direction that I wanted to go in. But I think as well, it made a lot of sense to me, I guess financially and also in terms of career progression, to step up to a PhD kind of scholarship level income while you have the kind of more freedoms and flexibilities of being younger, to then step into a career. I thought it would be much more difficult to step out of a career in maybe 10 years' time and go back down to being on a scholarship kind of income when you have probably more responsibilities around anyway. So that kind of logic to me made sense and kind of helped seal the deal towards choosing a PhD instead of going straight to industry.
Any other questions? Yes.
A lot of your topics are very specific. So the blink and like to me when you said, oh, I'm making the implant to allow someone to blink, it's so specific and something that I would never think that someone would be researching into that. How do you kind of come up and kind of lodge down, this is what I'm researching, or this is what I'm going to come up with?
It's probably, if someone was to ask what the biggest challenge of doing a PhD is, I'd definitely say that first 12 months of trying to actually decide what you're going to spend your time on and what's the sort of novel research contribution that you're going to make. It's definitely very challenging. Usually you'll work very closely because you're sort of starting out your academic career. This is the first time that you're sort of really thinking about, what is going to be my novel research contribution. It means that you'll be working quite closely with your supervisor, who's an expert in the field. And so they've sort of got a good understanding of, where the frontier of the latest, cutting edge research is. And so you'll work closely with them and they'll help sort of point you in the right direction. And then it's sort of having that kind of synergy of the master-apprentice relationship that sort of definitely will help you sort of formulate where you're going to really go.
In terms of the Blink project, so I'm working with surgeons from RPA who are head and neck surgeons. They're kind of, part of their specialty is removing tumours from the face region. So they see a lot of people who are in the position where they have facial paralysis, and a lot of the other interventions aren't working for them. So I guess they approached Greg to say, we need something to kind of fill in this space, and that's where I got involved. But it is funny that you say that Blink seems very specific, because to me it seems very, very broad, and I'm trying to work out what my specific questions within the PhD project are actually going to be, and what I'm interested in actually delving down into. Especially when I'm making this implant, I'm doing everything from understanding the biology to actually doing the CAD modeling and doing the printing and all of that sort of stuff. So it's very, very broad at the moment, but I'm only in my first 2 months, so I have to work out what I'm going to delve into and get that specifics.
Just on that, as a supervisor, when you know you're being successful at the supervision, or the student's going to be successful in their PhD, is if you get in a lift, let's say on the basement, and then you go up to the 4th floor, and by the time the doors close, you say, what are you doing for your PhD? And if the door's open on the 4th floor, and the door's open, you already know what they're doing, because they've been able to explain it to you in that 30, 40-second trip up there. That's when they're sort of really on track, because they can say, this is exactly what I'm doing, and this is what my contribution to the world is going to be.
If I could kind of add something there, I would say that it's kind of a mindset that comes with time. So I'm in my 3rd year of my PhD now, and I find myself like, you'll just be like kind of out in the world and something will come up and it'll be like, oh, I wonder if I could engineer something to fix this, or like, I wonder if I could apply what I'm doing here. And I found that kind of, all these new ideas pop up pretty frequently now, because you have that kind of curiosity and that experience in looking for problems and trying to kind of engineer a solution for them. So I guess I would say, even now in 1st year, just kind of keep your mind open. And a lot of times you can kind of develop topics that you're quite interested in just from the problems that you might see in the world around you, as you live it. So it's something that definitely gets easier as you go, but something that you probably aren't expected to like know now in 1st year. Like, I'm going to do my PhD in this specific topic, in this specific field at, this specific time. Yeah.
I think also relates to what you want to do with your internship. So when I was doing my internship, I work on coronary vessel heart disease in Concord Hospital, and that's when I know this is very interesting to me. So this only happened maybe like 2 years ago. No, not 2 years ago. 3 years ago, that I figured out this is what I want to do. And then I pursued my honors thesis with Professor Chinley, and then we started doing this project.
Just a show of hands, it might be because you're here today, but how many of you have actually given some serious thought to doing a PhD? Halfway put up? Okay. So we've got to do our marketing a bit better. So Ben had put up the dollar amounts, and I think it's still true. When I was getting the very lucrative $19,000 scholarship that I survived on with a family of 5, it was tax-free. Is it still tax-free?
It is tax-free.
Okay. So when you compare entry-level positions with a PhD, first thing, you don't go into a PhD for the money, but it's not that far off of what you would get as an entry-level person these days. It used to be pretty, get the ramen noodles out, and that was pretty much your feast for the week. And it's very different. The government has, I think, finally realized that that we're producing national treasures, not people that we want to just starve. It's a different way of looking at it these days. Any other questions? Yes.
With those awards, as you guys are referring to, the scholarships, how do you get into those awards, and what's the process into achieving those? It's not an easy step, I assume. What is the process?
Sydney.edu.au/scholarships. Yeah. Yeah. There's the one that Ben mentioned is, well, it's probably the most popular or prevalent one that the government offers, but many supervisors have their own scholarships through grants or other mechanisms. So it's not just what you see on that website. One thing that you can do is find out who's been successful at grants, because if someone comes knocking on my door and can save me 6 months recruitment process, I might look at you really quite favorably if you're really passionate and interested in the subject matter.
I think for the Sydney Uni scholarships, it's just one application form. So I know that in some places you might have had a similar experience if you looked at scholarships for undergrad. Some places might require you to submit separate applications forms for every single scholarship you might be eligible for. But with Sydney, it goes through one portal. So when you apply for your PhD, you simply tick a box saying, yes, I want to be considered for all of the scholarships that I'm eligible for. And you'll automatically have your name put on that list. But there are some external companies that will offer you scholarships. So I'm on a CSIRO supported place, and I had to apply separately through the CSIRO. So I had to kind of keep tabs on when their applications were open, and it was, pretty similar experience to what you would kind of expect for scholarships. Maybe a questionnaire and an interview. But otherwise it's pretty straightforward, at least for the internal ones as well.
Also, if you're an international student, it's also available to you. I'm an international student, and I'm also on the RTP scholarship as well. So it's not just open to domestic, international as well.
In the Faculty of Engineering, if you qualify to get a scholarship, they'll pay your tuition. So local students, that's waived from the federal government, but foreign students, they would otherwise be required to pay tuition, but the tuition is waived by the Faculty of Engineering if you qualify to enter into the PhD program. Any other questions?
What is your future goal or plan after finishing PhD? Are you willing to proceed a career as a researcher or work in industry?
Can anybody hear?
Did you want to repeat that? Just
I actually didn't hear it. I'm sorry.
What is your future goal or plan after finishing PhD?
That's a really good question. So in terms of future career plans, definitely right now, just trying to finish the PhD. It's just trying to get through it. But probably a big role model of mine would have been Dr. Ali Fadhi, who I was talking about in my presentation, who ended up making a startup out of his research that he'd done during his PhD. I think that it would be awesome to sort of enter the startup space and still sort of be collaborating and maintaining those academic relationships with the university. A lot of the time with the startups that are launched out of Sydney Uni, they still maintain those relationships with the university, those ties. For example, that bioceramic that I was talking about in my presentation, that was developed by Professor Harlis Reikat, who's the head of the ARC Training Centre for Innovative Bioengineering. Her research team developed that a number of years ago. and then they ended up licensing that patent out to Allegra Orthopedics, where Allegra Orthopedics is now wanting to commercialize it. But as they're continuing to develop that technology, it means that they still work closely with the researchers at Sydney Uni. So it's sort of they still sort of maintain those relationships. And so what it means is that you can sort of hover in that space between, researching in industry and with academia as well. So that's probably somewhere that I'd like to be in the future.
Since I'm new to the whole PhD thing, I'm still testing the waters about research. Hopefully, because I kind of see it as two different pathways. You can either stay in research or go into industry. So throughout my PhD, hopefully we'll find out if I like research, how much I like research, and then might look into doing a postdoc somewhere and getting into some other research, maybe another university, maybe here. Or there's always the option. I'm kind of hoping that by doing the PhD, I'm going to be upskilled so I can get into something that I would be really passionate about in industry if I don't keep staying in research.
Yeah, kind of the same as Jacinta. I probably stay in academia or just do more postdoc somewhere in another country or something like that, or just get into industry if I have the opportunity. So it's really broad. It's not, don't limit yourself to just academia. You can also do industry job as well.
Yeah, I'm almost 100% sure I'm moving away from academia at this point. I'm really lucky in that my field, there are a lot of kind of small emerging startups, especially in kind of San Francisco and Boston, and a few across Europe as well. There are some really kind of concentrated hubs of new companies that are kind of emerging with their kind of genetic engineering tools and technologies. So I would really love to step into kind of like an up and coming kind of synbio company and work in industry. But I guess the benefit of working for a smaller company or one that's still kind of growing and finding their feet is that you are still really heavily involved in the research work and in the bench work as well. So I like the idea of kind of helping to translate the science and the research into kind of a tangible commercial engineering, like engineered product. So kind of acting as a translator between kind of bench scale genetic engineering work and kind of commercial scale fermentation scale up.
You had a question in the front row. Do you still have it?
So is there a point in terms of your academic achievement where you would suggest maybe you shouldn't do a PhD or maybe you should proceed on doing PhD?
Let's see. Passion is the first thing. I would, if you go into a PhD because you feel like you can't find a job in industry or your mom and dad told you have to do a PhD, that's not a good reason. If you have honors, I can't remember what the cutoff is, but if your marks are pretty good, you can transition from a bachelor's to a PhD. If you need to sort of up your skills in the WAM department, then you can do that through a master's degree. Where would you? So your question is when should you not do it? Is that right? Yeah. So.
Is there a cutoff point-ish kind of thing?
You've just gone through do you remember the?
I think the entry requirements were around second class, high second class honors to first class honors is sort of that benchmark around.
Which is what, like distinction in undergrad terms, I guess, like around that 75 plus kind of mark. Yeah. But I guess as well, with your WAM and getting into programs and things like that, I would say that your WAM has more of an influence on how likely you are to get given a scholarship. So the RTP scholarships are pretty competitive depending on what field you're going into, and so they'll take into account your academic performance. So you might be able to get into a PhD program with much lower kind of average mark or a lower honours mark, but then you might face problems with getting scholarships if it kind of drops too much lower than that second class honours kind of bar. But it's possible you can self-fund your PhD as well if you really wanted to. I'm sure there are people out there who do. So that would be my only consideration with if your WAM is too low.
Yeah.
terms of collecting your data on like bone grafts or the limb, how do you go about actually getting these results, or like actually testing on animals or people?
So usually with biomedical technology, this is stuff that you're implanting into people's bodies. So you want to be confident that when you're going to implant this technology into someone, it's going to have a positive effect and not a negative effect on that patient. So usually what will happen is that whenever you're developing a biomedical technology, you're going to go through a series of clinical stages where you're trialing the product. So usually it'll start out with a sort of preclinical testing in which you're doing, computer modeling. So Dylan and I specialize in that component where you're sort of just simulating what the device would function as when it's implanted. And then what's going to happen is that you'll do animal studies, and then you'll move on to small stage human trials with 10 patients, and then you'll do a second stage human trial with around 100 patients or 1,000, and then it moves on to more humans, a larger pool of humans being tested. And so having that sort of those stages means that you kind of you're
being cautious.
Yeah, you're sort of being cautious. Yeah.
So would you just contact the hospital and just like ask them for maybe testing something else? How do you start it off?
So at UCED, well, obviously you're not going to be doing human studies unless you've done many years of work. In terms of my project, we work strongly with RPA, with some surgeons there. So in the future, if we were going to move into human studies, it would be through their contacts. But at UCED, there's very strong human ethics committees, animal ethics committees that kind of make sure that everything we do needs to be done firstly and is being done in the right way, so everything is as ethical as it can be.
And places like Sydney University have some pretty spectacular equipment and facilities. So Jacinta's project is taking place in the basement, I guess you'd call it, of the Charles Perkins Center, where they have this place called the Hybrid Theater, which is just spectacular as far as a research center. So when you are doing a PhD, you have access to the university, which is, I guess you'd say, billions of dollars worth of infrastructure that companies just don't have, and their jaws just drop when they come and see the stuff that we have here. There's a really special environment to conduct research. It's specifically tailored for getting to the point where you're saying, or we want to go from idea on a benchtop into a human use or clinical use or veterinary use or whatever. Any other questions?
Yes. I had one question. It's more about choosing what type of engineering stream you're doing. You hear a lot of people transfer from like mechanical or electrical engineering into like more biomedical aspects of it. I was just like asking, what are the perks of doing like a staying in the biomedical stream and then specializing in like bioelectronics, or going into the electrical engineering stream and just move like more doing like a lateral movement into like the biomedical field? So like, do you think it's better to be more specific in just doing bioelectronics, or doing a more broad subject base at the beginning and then specializing in bioelectronics?
I think definitely as you sort of go on in your degree, you'll learn more about what you enjoy, and then you'll end up sort of specializing in what your passion is. So right now you guys are in first year, where you're just sort of learning really the fundamental language of how to talk like an engineer. So, you've got to learn your maths, your statics, your chemistry, just sort of the foundations. And then what happens is, what I found is that as I progressed through the degree, my third and fourth years were a lot more sort of enjoyable because we were really applying and learning about biomedical technology rather than sort of those building up those fundamental skills that are required. And so what I found is that as I just sort of learned more, particularly in those third and fourth years, about the biomechanics, bioelectronics, those actual sort of specific sub-disciplines of biomedical engineering, it meant that just by virtue of learning, I was able to find out what I was a lot more passionate in, and then I sort of followed the direction, and in the end, I did the subject of tissue engineering. Absolutely loved it, and then that's what I knew that I wanted to spend my time on.
I guess one thing is that if you're interested in moving into this biomedical field, like materials in normal engineering are not the same as materials that can be implanted. Electronics are different. Like everything's kind of very specific to the topic. So if you do a more broad degree, so like if you do electrical, you still learn all the same fundamentals, but there's a lot of very specific things you need to know to be able to apply that to the human body. And I guess that's the benefit of doing the biomedical degree. So if you know that you're passionate about doing the medical engineering, then this gives you the good basis to kind of know how you can apply the engineering to the field that you want to apply it to.
I guess when I look back, knowing what you want to do is a bonus, but hardly any of us do know exactly what we want to do. But what we do at universities is hopefully teach people how to learn. And so I did mechanical engineering as my undergraduate degree, but quickly realized that transmissions and things like that wasn't my passion. I went into electronics, and because I had gone through an engineering degree, I actually could transition to do electronics. And then when I really found what I find my calling is working in neuroprosthetics, I worked for the company Cochlear for a while, and it was just spectacular. But it was the ability to learn. So if you think you know what you want, go down that road. You're not going to have regrets. You're just going to realize, oh, maybe I didn't learn the things that I wanted to learn, so I can now go and do those other things because you've learned how to learn.
Yeah, I guess one more comment on that is people have a really kind of strong tendency, if maybe you've committed you're 4 years into a combined electrical engineering degree, and you just realize that you really don't want to, like you wish you had done biomed or something. People kind of hang on to the time they've already spent on something, and they'll say, well, I'll just stick it out because I've already spent all this time doing electrical engineering. But I think there's like something to be said to kind of keeping your mind open to pivoting. So you can always pivot, and the time that you've spent doing one thing isn't wasted and it's not lost. The skill set that you kind of gain from doing things in the past that you may not be wanting to do now still kind of gives you a breadth and a base of knowledge that kind of sets you apart from someone else who might have taken a different path to get to the same end goal. So I guess don't be afraid to pivot at any point in your degree, your career, to kind of find a better path that feels more suited to what you're interested in. So I would say, stay on biomed now if that's what you're really excited about. And if you feel like you need to pivot later to maybe take a broader, electrical kind of knowledge base, then you can always do that when that need comes up. So try not to look on the things that you're doing now or look on time spent on something as being a waste, because it rarely is.
I'd like to thank the panel and give them a round of applause, please.
So you work in teams of people. How does that work? Like other teams of people, are they doing PhDs as well, or how does that work?
So the question is about working in teams and how it works within a particular laboratory. And I think it varies from laboratory to laboratory. Certainly in my lab, we have people that are doing what's called a postdoctoral position, where they have already received their PhD and they're working towards building their research career. We also have undergraduates that are working with us as interns. We have recent graduates and PhD students all working as part of a team. And they're not really, in my lab anyway, it's not really strict lines of demarcation. You might find the PhD student is helping the postdoctoral researcher and vice versa from time to time.
I might just add on to that. We also, it's not just the people we talk to in our team, it's also cross-team. So I'm actually in Alexander Kewen's lab, which is bioelectronics, but Greg Swanning works on the bionic eye, and his team is obviously very closely related to what I'm working on. So I'll frequently have discussions with people in his team just to see what they're doing and talk about my problems as well.
Also, just for my PhD, you know, I've actively sought out various clinicians and people with expertise in the field, and actively gone out and kind of, actually one of my supervisors, he had nothing to do with my PhD, and I realised he had a background with EEG and monitoring stroke patients, so I actually approached him, talked to him about my research and asked if he would like to come on as a secondary supervisor. So I kind of reached out to my own research and found people to come help me out as well. So you can kind of find your own people and stuff, make your own collaborations too.
And a lot of the time people will be working on stuff that's similar but like different enough that your projects are defined. So maybe, for example, you could work on an animal study together and both get data from it, because obviously that makes it much more useful. But technically you're looking for asking different questions but using similar samples. That's another way that you can work with other people.
And engineering, if you read an engineering research paper, you'll see that there's like 8 authors on it compared to, you know, like a social science research paper that's just got, you know, 1 or 2 authors in it. The reason why engineering research papers are have so many collaborators on it is that it's a very multidisciplinary field, and in order to produce research, it does require a collaborative effort. And so what you'll find is that within the biomedical school, there's many different people with different specializations, and you really create a nice synergy with that. Yeah.
Did that answer your question? Yeah. Any other questions?
Yes. Are there any tips for first-year students who are looking into doing a PhD?
Are there tips for first-year engineering students that are seeking to do a PhD? I would say stay the course. You're not doing anything wrong if you're not preparing for your PhD at this point in time, other than pursuing your passions. The last session we had earlier today, we were talking about pivoting. If you're going down a particular pathway that you're not a great fan of, don't worry about pivoting. I've pivoted 3 or 4 times in my life, and it hasn't hurt me in any way. And in fact, I've enjoyed the experience, so I wouldn't, maybe other people will differ, but I think do your undergraduate degree, enjoy it, and find your passion.
I think Ben put quite a good point in his slides. I think a few of you would have remembered, but it said tailor your electives. And that applies to both pivoting. If you're not enjoying one, you can change to others. Especially in your final years, whether you're doing a double or a single, that might be your the third, fourth, and fifth years of your degree. I would really suggest looking at the electives which you really enjoy and you want to learn from. So maybe a elective is not particularly what you want to be doing, but it might be very beneficial to learn from it. In the end of the day, once you get to your honours year, and there'll be a list of projects which come out, you need to take that initiative to talk to the supervisor to say why you're best and why you should be doing that project. And don't be afraid if you end up not being able to do that project. There may be another project which you may actually really enjoy and may be a good thing. I think that's probably happened with most of us. We didn't get our first choice or project, or the project changed halfway through, and that's good. Change is always a welcome thing.
Also, I'd say definitely just put yourself in a position where you're going to be able to expose yourself to as many different areas of research within biomedical engineering, because there are so many different areas that you can go down. And what you'll find is that right now you're just in first year, so you're just learning your fundamental building blocks, you know, sort of how to speak like an engineer, you know, doing your maths and your physics and your chemistry and all of that. But what will happen is that you'll find as you go on through the course, you'll be able to develop, or you'll be able to learn a lot more about actual specializations of biomedical engineering. The other thing is that I would definitely recommend with your internships that you do throughout the summer, that's a really good opportunity to expose yourself to, the frontiers of cutting edge technology. And there's, great opportunities with, biomedical companies just in Sydney where you can do that. The other thing is with, startup companies, you can do a internship with a startup company. So Dr. Ali Fadhi, who I was talking about during my presentation, I did my two internships there. And what it meant is because it was a startup, every single day you're being put on a different role, and it meant that you actually I exposed myself to a very wide degree of different roles. And so it's definitely, you'll sort of find, like right now you sort of don't know what you don't know, and so you don't know what you like. So it's sort of, as you sort of, go on further and put yourself in a position where you're going to be able to learn more, you'll find out what you like and develop your passion from there.
I think my only other bit of advice for your question of, as a first year that's keen on PhDs, is just ask questions. You have really great tutors in all your subjects across biomedical engineering, and they are all PhD or postdoc students that have gone along that journey. Your lecturers as well, they're heads of research labs. Ask questions, find out what people are working on, what projects exist, and yeah, just stay interested, I guess.
Any others? How about up in the back? Any? Don't be shy. Yes?
What does day-to-day look like as a PhD?
So what day-to-day looks like, that's a really good question. PhDs are awesome because it's really different. So some days I might go into the lab and I know I really need to run an experiment that day, and it's a day-long experiment because it's got lots of incubation periods. Other days I might go in and go, you know what, I haven't read a lot of literature recently. I'm going to really deep dive into, what papers have come out recently. Or I might have teaching, and we come and teach you guys, and that shakes things up a bit as well. But the great thing about a PhD is like you are sort of your own boss. Like your supervisor is your boss, but at the end of the day, you're project managing your project. It's on you to make sure that you finish it within the 3.5 or 4 year deadline. So if you go, you know what, today I'm going to take it a bit easy, or today I'm going to absolutely drill in really hard, stay here later, it's totally up to you. And that's what's really, really cool about a PhD, because no one's clocking you in or out.
I would just really echo what Matilda was saying, is that it's a very dynamic job, because every day is a different day. And so I think that's a great thing, is that you're always in a position where you're learning something new, you're applying what you learn, and it's a very sort of stimulating type of thing. And so, yeah, I'd definitely recommend it. And I think you get a lot of satisfaction out of it as well. Yeah.
And another aspect of it is what the 4 PhD students you have here are doing today, is expressing and spreading the interest in the work that they're doing. It's a huge part of what a PhD is. It's not just doing the work, it's sending it out to the world through academic publications. That distributes it throughout your community, but I think there's probably not a week that goes by that you don't have some opportunity to present or go on a lab tour with some high school students, et cetera. So I think that part of it is, I would rank it in the top 2 most important things, aspects about PhDs. There was a question over here, I think. No?
Same thing.
Oh, okay. All right. Anyone overcome their shyness in the back and want to ask a question? Okay, perhaps I'll ask one. How do you get involved when you're a PhD student? You're fresh out of your undergraduate, or you've just come back from industry. What do you do to become involved and be part of the biomedical community at the university? sorry.
Sorry, but I'm cutting you out. I guess the way I sort of got involved was we had weekly meetings for most of our supervisors and was really listening to what others are doing. So learning how their process went, talking to them, and then also trying those things ourselves. I think for most of us in our PhD year, the first thing we did in our year was do a literature review. And by now you will be writing your literature review, which will be due soon, which is great. And you do a few more of those throughout the uni. You know, you write your literature review in your honours year, you think it's great, and then you read back over it and you don't want to publish it. So I guess you always have something back to follow on to, so literature review, but it gets a bit tedious at times. So for me, I work in 3D printers, so I sort of tinker on them, write the software, play around with them. So that was me, but it was also just really listening to what your what your fellow PhD students and postdocs have been doing.
I think one other thing is, like, I used to be part of the biomedical engineering community in terms of, I did my undergrad through there. And obviously I've moved over to medicine since, but it's been really great still having the connections with people like Ben and Jacinta, who's been in one of the earlier sessions, because they're people that I did my undergrad with, and then also through teaching, I've met other great PhD students like Sam and Jet. And so you can sort of get involved by not being afraid to sort of have a chat after a tutor meeting and sort of, yeah, like I've been so amazed hearing about what you guys are up to. But yeah, you can get really involved in the biomedical school because everyone that works in it's pretty friendly and collaborative, and there's sort of lots of different research groups. But also people like Hala, who you guys may have come across, run some really great initiatives, and getting involved in those has been another great way to meet people in the biomedical school and get involved there as well. So that's other stuff that you can do to spice up your day-to-day as well. And
I'd also just touch that I'd really recommend be proactive. There is nothing that I think would impress an employer or a supervisor or anyone when a student is reaching out, sending an email, going, "I've checked out some of your research and I think it's really cool, and I'd love to be a part of the research program that you're putting out." And in the end, the whole point of an initiative such as this is that we're really wanting to engage with you guys, and a lot of the time with many academics and professors, they are people, and there's nothing. So I definitely recommend approaching them, talking to them, and taking an interest in their research. And so, yeah, being proactive is something that I definitely recommend.
Yes.
Did you ever feel like burnt out after you did your undergrad and honours and then moving straight into a PhD?
So the question's about burnout. Did you feel burned out after finishing your undergraduate and before you started your PhD? Just one answer, one-word answer, probably.
Yes. So I did a 5-year degree, and I went straight into a PhD because the opportunity was there. If I didn't take it, I probably wouldn't have got it. Someone else would have taken it. So I took it. Great. And then COVID struck, and went to work from home, and I was just super unproductive until luckily I could come back into the uni and work, and that got me back into it. But yeah, it happens.
Yeah, I had a slightly different journey. I took a year off after my undergrad degree, and I did some work in a random job in an office for a year, and then I went travelling for a year, had some overseas experience, and I came back and sorted out some kind of further research and got into a PhD that way. So if you're worried about burnout, I think travelling and that kind of thing is obviously a great opportunity to get experience, things like that. So a good way to break up that burnout.
I have a similar experience to Sam. I actually took a year off as well because I wasn't 100% sure that I wanted to do a PhD. And I would say that it's not a small commitment, right? You're committing to something for another 4 years. And obviously if you're doing a PhD, you love research, you loved your undergrad. So it's definitely something that you're interested in, but I needed a little bit more time, a bit like Sam, to figure out that I was ready for another 4-year commitment. And I would recommend it, but I think Jed is also right. Sometimes momentum on some projects doesn't allow for it. But just be aware that you're human and you're allowed to take breaks. You do get holiday allocation as part of a PhD. You're not expected to go sort of every single day. And that's really important for not getting unwell, a bit like honours years and school and all that sort of stuff. So look after yourself is my big bit of recommendation, but also pace yourself. Know when you've got to work really hard and know when you can take it a bit easier, and talk to your supervisor about it as well. Be really honest with them, because it's a collaboration at the end of the day with your supervisor. And so the more honest with them you are about your mindset and where your head's at and how you're feeling, the less likely things like burnout are going to really harm you, because it's more likely that you've got the support that you need, that they'll sort of turn around and be like, you know what, maybe take a break or come back to it with fresh eyes. Because yeah, it's a marathon, not a sprint, is what they say. And taking the time to make sure that you're looking after yourself and you're doing it right is awesome. And the supervisors at UCIT are very understanding and a great team, so yeah. Do you want to add anything?
Oh, that was great.
Any others? Yes.
Do you have part-time jobs to compensate other ways?
The question is, do you have part-time jobs to offset the stresses? Aha. Yeah.
We do this.
Yeah,
we teach you guys.
It's called teaching. So teaching is really rewarding. I actually also teach cello at home. I was actually going to be a professional cellist. But then I chose engineering because I loved it and I hated practicing. But yeah, we teach as well. And it's really, we actually do love teaching and giving our knowledge and helping you rise up to become the next sort of set of engineers.
I also used to do debating coaching until quite recently as well. Just, spice it up, throw in something different. But also with a PhD, you do have time for hobbies. I still play team sport with my friends, join choirs and things. I think it's important to keep balance, right? All through school, they all recommended that you do lots of extracurriculars. And I don't think that becomes any less true the older you get. So I would highly recommend, like you say, taking the stress off by diversifying what you're doing, but do make sure that you're leaving enough hours in the week for your PhD. But as long as you're doing that, then there's plenty of time for hobbies, I think.
And the other option is that you don't have to do a PhD full-time. You can do it part-time as well. So there's some research PhD students in my research team who are doing it part-time, and then they're actually working at a company that sort of has interest in the PhD. So many students who are they'll be working out in industry, and then they'll go back and have the industry sponsor their PhD. And so what they'll do is, 2 or 3 days a week they'll be working at the university doing their PhD, and then the other days they'll be working at the company. So you don't have to do it full time. You can also do it part time and work in industry.
Yes.
How many years is the part-time PhD?
It's basically double. So you go half-time. Actually, I've never tested it, but I think
8 probably.
Oh, sorry.
7 to 8.
6?
6 to 8.
6 to 8.
Yeah.
Oh, yeah. So basically double the time. I've never tested it, but you might be able to do something like a 60-40. I'm not sure about that. If you had 2 days a week, you're working somewhere, you might be able to do your part-time as or some sort of pro rata thing.
One thing to note is you're sometimes not eligible for some scholarships if you're part-time though. So the benefit of being able to work more means that essentially a lot of the government scholarships, it's sort of like you're not eligible because it's not your full-time gig.
Is it possible that you end up not getting anywhere in your research plan?
There's this famous story about Stanford is usually the example that people use, where a PhD student has just finished their thesis and they're just about to hand it over, and word comes from afar that a publication has just come out and it's completely gazumped the person that's handing in their thesis. I have heard of it happening, but it's always a friend of a friend. I've never actually seen it happen. What tends to happen is you change tack a bit. If someone has made a big discovery that you were on the verge of, you did get beaten to that, but you change tack a bit. Very rarely does that happen? I mean, basically, a PhD is a directed research program that, independent research program with a mentor that the objective is to contribute to knowledge and learn how to become a researcher. And if you fulfill those requirements, that's the most part of it.
But also in terms of potentially not finding results, sometimes you do get null results, but they're results too. Sometimes it's actually worth publishing that something didn't work, because then hopefully other people won't waste their time doing it again. Whereas if you don't publish that it didn't work, inevitably somebody else comes along and goes, ha, gap in the market, let's give this a red hot go. And if you just have everyone perpetually repeating this experiment that doesn't work, that's a disaster. So null results are still results, because it's still, you've tried a method or a tack, and finding out that that's no good is sometimes just as important.
I think also it's very common from my experience, from other PhD students I've spoken to as well, is that you'll start your research down one avenue, and then as you guys persist during that first year period, you realise you need to kind of change it slightly. So you might, whether it's through the literature review or whether it's through studies, you realise you need to change tack slightly. I think that's very, very common with PhD students.
I might also add that even though you're doing a PhD, it's not like you have to make a commercial product out of it. You don't have to. It's not a requirement. It's just a lot of us like that approach and we would love to. But in the end of the day, if it doesn't become a commercial product, we've still contributed to knowledge. We still have a huge amount of knowledge we've learnt, and we can go forth with that knowledge. And I guess what Greg said before was pivot. So you can use that knowledge to make something else, which is great.
Do you mind if I ask a question?
Please.
So Sam, I was just really interested about your startup. Do you mind telling us about where you where you want to take your research in the future, and just a little bit about the startup gig?
Yeah, sure. So, yeah, like I said in my talk, I have a patent that I filed a few years ago, and going down that patenting process, I realised I should create a startup company. So I've got a startup company called Neuroflux, who owns that patent. My day-to-day is really focused on the research, kind of figuring out whether and how valid this method is, if my device is going to be effective. So as I progress my research studies, I'm also developing my startup company along the side, just slowly, so that if my research becomes viable, I can take that forward. So I'm actually currently doing an accelerator program through UNSW that kind of helps me give a bit more insight into kind of business models and pitching and all these different kind of skills I don't get during my PhD. But yeah, my goal, if everything looks good, I'd like to take my startup company forward and make a commercial device out of it. But my research and PhD is really coming first. I really want to get my PhD. But I'm just wary that there is a potential for commercialization, so it's really exciting to kind of build that startup on the side just in case it becomes viable.
The short answer is yes, but there's probably...
I'll just repeat the question for the camera. It was pretty much saying, can you use other companies, so other than those suggested to you? For myself, I work with osseointegration, which actually implants abutments in the arm or leg to use for prostheses, which is great. I also work with other surgeons as well, and I also am in the process of looking to get some experience from another company, not necessarily saying, hey, I want you to use my research, but just learn from them. So the answer is yes, we are allowed to reach out to other companies, but it comes down to us. So your supervisor may suggest a company, but they're not going to be the one who are going to get that link. It's up to the PhD student. If you feel you need more companies, then go for it.
And also, the ARC Training Centre for Innovative Bioengineering, that is sort of a centre that really wants to encourage and maintain relationships between academia and industry. And so in many cases, when researchers, like biomedical researchers develop some technology, they may license that out to a company, but the company, when they're going to commercialize it, they need to still continue developing that product. And so they'll still maintain those linkages with the researchers at the uni so basically the researchers continue working on, you know, the product, and then the company sort of ends up commercializing it. So it's sort of like, I think it's sort of a good thing to emphasize that the path of academia, particularly in biomedical engineering, is not just self-contained to, you're not just sort of, you know, in your own little kind of room just working on your theoretical like problem. It's very much, you know, we're really wanting to encourage industries. And so in many cases, to answer your question, in many cases, industry will be very interested, or can be interested in wanting to have a collaborative relationship with you.
Can I add to that? Just to add to what Ben was saying, one way that industry or commercial companies and things might become interested in your research is through something called conferences. So I don't know whether you guys are that well versed on what a conference is, but essentially various research areas have different sort of annual conferences where essentially people that are researching in those areas get together and present the work or the most recent findings. And that's actually a really exciting place for one, as Greg was mentioning earlier, PhD students to sort of communicate their ideas. But it's also a really great place to sort of network and make those sort of industry relationships and get people really excited about what it is you're discovering. And one of the great benefits of being a PhD student is a lot of the time those conferences are overseas. So can't wait for COVID to be over, because hopefully that'll come back. But that's something else that's really great about a PhD, lots of travel opportunities.
Yeah, from personal experience, I'm a bit older than these guys, but I think I've been to 75 countries. Not all of them because of conferences, but every year there's a conference when there's not COVID, and they very rarely have them at you know, sort of the place that you don't want to go to. There was one at a shopping mall in Dallas, Texas, but that was a standout. Everything else was like the beach at Cancun, Mexico, and Argentina, and all sorts of fantastic places. And nearly got struck by lightning in France, but another story. But all of this stuff is part of the deal. You know, you go into academia, you do have a lot of freedom.
And even in a COVID world, the interstate ones still exist. So there are still travel opportunities, just at a more domestic level.
Yeah.
And Zoom, of course.
Yeah.
So.
I'll just emphasize, absolutely reach out to your tutors and your professors and different people around the school. I really recommend it. Yeah.
And add us all on LinkedIn if you want. And same with the industry reps that you guys saw on Monday. Know most of them personally, and I know that they'd all happily add you on LinkedIn and answer any questions after these sessions if they pop into your head or you're preferring to ask questions in private.
And there's also, make sure you join SUAB if you haven't already. If you haven't, I don't know, come on, join. It's $5. And they have a SUAB cocktail night, which is coming up in a couple of weeks. I would actually really recommend as a first year to go along and just have fun and talk to people, because mostly the industry members are barraged by third-year students who want an internship. And they all would love to talk to a first-year student who just wants to learn and talk to them about what they're doing. And it also gives you some exposure. You get those contacts, you put them in LinkedIn or your mobile phone, and come second and third year, you already have some people who you know, so you're not afraid and thinking, oh my goodness, I've got 1 week to get an internship. What do I do? So,
and the best networking comes from someone enjoying having a conversation with you, rather than you walking up to them being like, what job have you got for me? Tends to not work.
Thanks, everyone. Thanks for your good questions.